My name is Sean Joseph. I'm the Director of Pharmacology at Calibr. And I'm excited today to talk to you about one of our more advanced programs. And this is all relating to a regulatory T-cell approach to Parkinson's disease. So Parkinson's disease may not have struck you first as an immune disorder, but essentially growing evidence over the past 10, 20 years has pointed to inflammation, to the progression of Parkinson's disease and other neurodegenerative conditions such as ALS, Alzheimer's disease, and others. And neuroprotective therapies in particular that slow or reverse the neurodegenerative process are urgently needed for Parkinson's disease, as well as these other neurodegenerative diseases that I told you about. And to kind of give you a cheat, this is my cheat sheet for the talk. Essentially, the evidence over the past couple decades that has really pointed to this being a good idea, upregulation of Tregs, is that preclinically, GM-CSF treatment has shown to modulate innate microglial immunity. This increases these regulatory T cells, which are anti-inflammatory in nature. These cells then transmigrate into the brain from the periphery, and this results in anti-inflammatory as well as neuroprotective responses directly. And clinically, it's actually been shown by Howard Gendelman's lab at UNMC that treatment with GM-CSF peptide, also known as sargramostim or leukine, results in increases in Tregs, and those increases in Tregs then improve motor function, have been shown to improve motor function in patients with Parkinson's disease. But the big issue with this treatment, leukine, is that it has a very short half-life, and these are Parkinson's disease patients, and it's hard for them to self-administer this drug subcutaneously. And so a long-acting GM-CSF would likely increase compliance in the Parkinson's disease patient population, and this could lead to fewer side effects overall and greater efficacy. So we applied our Calibr half-life extension platform, which I'll talk to you about in a minute, to essentially extend the life of that GM-CSF peptide. And we've now developed this into a novel immunomodulatory therapy for Parkinson's disease, but also potentially for Alzheimer's disease and ALS, as well as other neurodegenerative diseases. It also turns out that GM-CSF peptide, or leukine, was recently approved for acute radiation syndrome in humans. And so this actually could be a great countermeasure molecule as well for radiation exposure. So some of the preclinical data that I alluded to essentially are shown on this slide. It's a busy slide, but essentially what this slide is meant to tell you is that across multiple Parkinson's disease animal models and Alzheimer's disease animal models, GM-CSF treatment upregulates Tregs, it enhances their activity and their function, and then it also then reduces neuroinflammation. And this essentially is shown across many different models. You can see here that there's a pretty dramatic effect bringing, with treatment, GM-CSF back to control levels of neuroprotection, or dopaminergic neurons are essentially fully preserved. And in Alzheimer's disease models, you can see here that you get essentially reverses the cognitive decline that occurs in Alzheimer's disease animal models and reduces the amyloid burden, which is a kind of a pharmacodynamic biomarker of the disease progression. So interestingly for ALS, and this is a little bit of a side, but I thought it was important because it generally speaks to the idea that Tregs and Treg upregulation, or expansion of numbers as well as improvements in function, can affect multiple neurodegenerative diseases. Essentially, someone recently took T regulatory cells that they've harvested from patients. They put those Treg cells into those same patients that have ALS, and essentially completely suppressed the progression of the disease. And interestingly, when those cells essentially faded away, the disease dramatically comes back, and you can then stunt it. They've done 4 cycles, and they were able to slow the progression each time. So interestingly, this is a potential therapy for ALS as well. And we're pursuing this kind of at the preclinical level at the moment. So I mentioned this earlier, that this phase 1 placebo-controlled clinical trial run by the UNMC lab with Howard Gendelman essentially kind of built on the idea that in Parkinson's disease, it is a neuroinflammatory condition. And in this disease state, you get a dysfunction of regulatory T cells. It's not only the numbers decline, which they do, but you also get a decrease in the function overall. And so based on all of the preclinical data that has been shown with GM-CSF, he thought this would be a good idea to essentially put this molecule in humans, which is an FDA-approved drug, sargramostim. This is the wild-type GM-CSF peptide, and showed an increase in these patients of increase in Treg numbers, increase in Treg function, and this correlated with dramatic improvements in motor function, and in these Parkinson's disease patients essentially getting their independence, regaining their independence. He talked about patients who had essentially couldn't mow the lawn anymore, couldn't drive, and suddenly they're able to do those things they weren't able to do in the past. So this led us, again, we have this problem with leucine or sargramostim in that it has a very short half-life and it's hard to self-administer for a Parkinson's disease patient. And there are some side effects that are driven by the high doses that are required. So using our long-acting technology, we essentially built a long-acting GM-CSF. And to sort of gloss over maybe 10 years of work done at the Scripps Research Institute even before Calibr came on board, essentially there is a discovery at Calibr showing that there's a bovine antibody that has this really interesting globular domain at the end of the antibody. Turns out with, again, a lot of work that was done by scientists before I had come, essentially showed that you could remove this globular domain and you could replace it with a peptide of any type you'd like. So GM-CSF, in this case, we've done other molecules in the past, and you're able to then essentially get the wild type potency of that peptide, but you get the extended pharmacokinetics and the extended efficacy or pharmacodynamic effects. Now, so we use this technology to make a long-acting GM-CSF. And this, again, to summarize, a lot of work in terms of optimizing expression levels, optimizing the immunogenicity of the molecule, removing antibody-dependent cellular cytotoxicity, and so forth. It took a decent amount of engineering, but in the end, we got a molecule that you can see here had the activity similar to GM-CSF. This is an in vitro assay. And then had the pharmacokinetics that are more like an antibody than GM-CSF. We took this molecule and we've now put it into rodent models. We've put it into non-human primates. And we're able to show that you get, with a single injection, you're able to increase those T regulatory cells. And that lasts almost as high, peaks somewhere at 20-fold higher than normal levels. And it lasts for actually almost 2 weeks. And importantly, in the non-human primate especially, this molecule is very well tolerated. So we put this molecule into preclinical animal models for Parkinson's disease. And as you can see here, we got a nice dose-dependent increase in T regulatory cells. With data not shown here, we've been able to improve their function with our long-acting GM-CSF molecule. And then we were also to dampen that inflammation that occurs in the brain, mainly as read out by microgliosis here, and then a protection of dopaminergic neurons, which are normally affected in Parkinson's disease. So here we are at this point. We've done a lot of the early clinical development, the preclinical development, and we're currently in the IND-enabling phase. And at the moment, we started on February 1st in manufacturing of this long-acting version of the molecule. And with biologics, as many of you may know, take quite a long time to manufacture. So at the moment, we're looking at clinical entry in 18 to 24 months. And so at the moment, we're designing the clinical trial for the phase 1 in healthy volunteers and with a cohort population from Parkinson's disease patients. With that, I'd like to conclude and thank all the people who actually made this possible. This is a small group of people who were associated with the project. And as I look at this list, I realize that Feng Wang, who actually made the molecule, is not on here. So I want to make sure that I acknowledge him and all the critical contributions he's made.